Hello, everyone. Good morning, good afternoon, and good evening. Welcome to Life Science Across the Globe. My name is Janine Stevens, and I am one of the Janelia organizers of this series. Today's event is focused on sustainability, hosted by Cold Spring Harbor Laboratory, and this is our last seminar of 2021. We do aim to resume in February, and we hope to have a full lineup of events to share with you early in the new year. So today's format includes 3 short talks, followed by a brief moderated discussion, and then time for audience questions. Please go ahead and write your questions at any time in the Q&A box at the bottom of your screen. We will then conclude with the Meet the Panelists session, and we invite students, postdocs, and other trainees to stay on the call and to ask questions to our panelists and hear from them on various career development related topics. I also encourage all of you to please complete the brief survey that I will share via a link in the chat box, because your feedback is really important to us, and as are your ideas for future topics for the series. So please keep an eye out for that. I now want to turn it over to Rebecca Leshan, Executive Director of the Banbury Center at Cold Spring Harbor. Rebecca actually gave a beautiful talk in the Life Science Across the Globe series back in September 2020 about the unique nature of the Banbury Center and the impact of their meetings. And today she will make some opening remarks on behalf of Cold Spring Harbor Laboratory. So Rebecca.
Thank you, Janine, and thanks to everyone for joining. As she mentioned, I'm Executive Director of the Banbury Center, and on behalf of all of us at Cold Spring Harbor Laboratory, I want to welcome you to this edition of Life Sciences Across the Globe. Today's theme is sustainability, an issue that's close to us at Cold Spring Harbor, and one we hope is front and center on the minds of everyone around the world. Before I introduce today's moderator, I want to make a quick note of the image behind me, a typical scene at a Banbury Center meeting. But this one, a 1980 meeting focused on plant biotechnology, is not quite random. Sharp eyes will spot Barbara McClintock over my shoulder, and next to her in the striped sweater, our moderator today, Professor Marc von Montagu. Professor von Montagu is chairman of the International Plant Biotechnology Outreach Initiative at VIB in Ghent University, and he's one of the founders of modern agricultural biotechnology. I could spend a very long time going through his many achievements, honors, and awards, but I want to get straight to the panel. So without further ado, I'll hand over to Professor von Montagu to open today's panel. Thank you.
Okay, thank you very much. So it's a great pleasure to see the younger generations again. And now I think we immediately will start with Sarah Hearne. She represents the international organizations for plant research that was started in around 1940 in CIMMYT with the Rockefeller Foundation, and the grow out to the what's called the CGIR. And now we will hear from her, who is in the principal investigator for the corn and wheat breeding, how the work is going for the finding in the varieties, the wild varieties, the genes that all plant breeders dream of. So please, Sarah.
Thank you, Marc. Okay, so I want to talk to you today about sustainability, and in particular, what the contribution is of the international public sector breeding, specifically that within the CGIAR. So the CG was formed as an entity in 1971, before many people here were born. It's a global partnership of 15 research centers across the globe that collaborate to advance the agricultural development agenda. Now, very recently, we formed a new entity, which is called 1CG, where 12 of these centers are coming together to combine under one umbrella with a new research agenda that really looks at broader partnership to drive agricultural development, particularly focusing on the Sustainable Development Goals. So the SDGs, so we're talking about sustainability. There are 17 of them. You can go onto the SDG website to look at the UN goals. In particular, the work of the CG focuses on larger effect things like by 2030, ending hunger, ensuring access by all people, in particular those in poor, vulnerable situations, access to food and other resources. So the CG works in 5 main areas of the globe, including Latin America, the Siwana region, Western Central Africa, East and Southern, South Asia, and the Southeast Asia and the Pacific. We work across 5 main impact areas, which are outlined here on the screen. And what I want to talk to you today about, in particular with the SDGs, is the impact that breeding can have towards poverty, hunger, and climate adaptation. So at the moment, we have around 690 million people globally. That's about 9% of the population who are hungry. This information came from the 2020 State of Food and Nutrition report from the FAO. Now here I'm giving you maize and wheat as an example. We could scale this across other species, other commodities, staples like roots, tubers, bananas, other legume crops. But essentially, we have an increasing demand for these commodities. This demand comes not only from population growth, but from changes in diet. And the projected demand is here. This is the demand projected for wheat and maize by the FAO by 2050, so a big increase in demand. If we take the current trends of productivity improvements, they're seen here in the dotted lines underneath, we're not going to meet the demand by 2050. The additional challenge that we have is we've got disease, we've got the impacts of climate variability, climate change, we've got an increasing competition for resources such as water, nutrients, energy in agriculture, which all act to push this curve down. And what we try to do within the CG is devise new tools, new methods, new approaches through agronomy and through breeding to try to push this curve back up again, to try and meet these needs and demands. So if we look at climate change specifically, and I just pull out two factors that we all hear commonly about, which is heat and drought. Here is some analysis from 2011, but from David Labelle's lab, where he took historical data from 20,000 yield trials that we'd run in maize across Africa. And extrapolating from the data from the yield trials, for every degree day above 30 degrees C, we get lower yield under both optimal conditions, conditions that we would all like to see farmers having, where you have sufficient fertilizer, you have very, very good soil husbandry methods, you've got enough water availability. Even there, we start to see a decline in yield under higher temperatures. And under drought conditions, that effect is even more acute. Now, when we look at the germplasm base that we have for breeding, many of the current donors for droughts and the key parental lines that we use in the development of new hybrids are susceptible to drought stress at elevated temperatures. So we start to get a challenge of combined effects of these two particular stresses, which are challenging yield and causing a lot of issues when we look at crop production in Africa and beyond Africa. So this isn't just a textbook example. This is what drought looks like in the field. These are examples coming from the BBC. We see this when we work with farmers ourselves in Zimbabwe and other regions. Drought impacts food security massively, food availability. You get added effects here. You start to get issues of disease coming into these ears, aflatoxin, other challenges, all piggybacking on the impacts of drought and heat stress. if we look at maize yield projections, if I start this video, this is a recent analysis that came out earlier this month in Nature Food. There's been quite a lot of publications looking at climate change coming out recently on the back of COP26. And what you see here is a challenge pretty much everywhere in terms of maize yield projections as we move forward this century. Apologies to anybody who's red-green colorblind here. This is not my video, not my color scale. Those who are colorblind, most of the things are turning red, which is bad in this regard. So in many of the regions where we grow maize, either as a feed crop, which is predominantly in the US, and as a food crop, which is the predominant use in the global south where we tend to operate, we see a significant impact of climate change on maize yield. So just to give you a quick overview of what CG breeding looks like. The core of the breeding really focuses here on population improvement. And population improvement really is a closed population of elite by elite crosses where we drive hard and fast those populations to get as much transgressive segregation as possible. And from these populations, both in the CG and within our NARS and private sector partners, we start to extract materials that go through variety testing. Now, all of this is guided by farmers and consumers through the development of product profiles, which describe what variety should look like, and there are breeding targets. Now, within this, we will get lists of things, lists of ingredients that we need in terms of traits in product profiles that sometimes we don't have sufficient variation for within the existing elite pools that breeders are using. When this occurs, we start to look at donor and development, trait development. New donors that we can use to introduce new variation that's of value for specific breeding targets. Sometimes these donors exist in other germplasm pools, we can pull them in. Other times we have to go through trait mining. And what I want to focus on is that particular aspect of trait mining which relates to the germplasm banks and the gene bank collections of the CG. The CG holds 11 gene banks globally. They're the largest and most frequently accessed network of public gene banks globally. And we hold just over 750,000 accessions of many crop species, which are essentially the staples of the HHMI diet. And this is around 10% of the total ex situ collection held globally. 94% of all of the germplasm distributed globally in alignment with the International Treaty on Plant Genetic Resources for Food and Agriculture is distributed by the CGIAR. And of that, around 70% comes from CIMMYT annually. So wheat and maize going globally to a series of partners. Now, the question when we look at germplasm banks, this is the beauty of the diversity that we see. Now, that taken, most breeders would not touch this germplasm with a barge pole. This takes you back years in terms of your population improvement. Challenges of adaptation, other linkage drag. Not all diversity is good. Breeders have done a fantastic job of getting rid of the things that are deleterious, but there are some things that haven't passed that bottleneck. And the question is, how do we move forward and make the evolution and this broad diversity that we see work for us? So a few years ago, we redeveloped a concept which we now call environmental GWAS. And now we all know what GWAS is in a broad context. And here, essentially, we change the response variable. So with a standard GWAS, you have a genotype, you have phenotype data that you collect. You run that through a series of analyses to identify associations between SNPs and traits of interest. What we do here is essentially switch out to this phenotypic variable and use a different kind of data set. Data sets more relevant to the collection environments that landraces come from, that these accessions come from, to identify associations between SNPs and variables of interest. So what do I mean by these variables? Here we extend and expand the characterization information. So in the Maize collection, we have around 17,500 accessions that are collected from sites, and we know those geolocation points. We know where these materials have come from. Using the geolocation data, we are able, through the use of GIS tools, to extract short, medium, and long-term climate variables for those collection sites. Information like temperature, maximum temperatures, precipitation. And we're able to right-size this information so we capture the growing season in which the crop would have been cultivated, and other important metrics like grain and flowering time. Yeah, those periods which are most impacted by heat and drought, at least in maize. Now, we piloted some studies feeding on the back of another GWAS panel that we had, which we had actually phenotyped in the field. This panel was derived from land races, and about 2,700 of the accessions in that panel had collection site data. We took that data and we started to run it alongside in the analyses with the genomic data that we had. And this is one of the early figures that we had using precipitation. And as you can see, you can get very, very clear signal in terms of alleles associated with variation in precipitation in the collection site environments. This one happened to sit right on top of a heat shock transcription factor. Now, this early data, bits of it were published by Alberto Romero, a PhD student we had in Ed Buckler's lab. And the question was, how did we take this further? Was this really relevant in terms of something that was a target that was potentially breedable? So working with Jeff Rossabara, we've been crunching this data through further. So here you can see Manhattan plots looking at temperature, precipitation, isothermality, so the difference in daily fluctuations in temperature versus the standard oscillations you'd see annually, frequency of wet days during the growing season. And in all of them, yeah, we can get some really nice signal. When we look at the top 1,000 environmental GWAS hits that we get here, and we start to look at the phenotypic response, this is looking at yield. Yes, we see a difference when we look at SNPs that are selected under or that come from that particular analyses versus random SNPs. And when we look at yield here under low precipitation, yes, the candidate alleles that we find under low precipitation do enhance yield under drought. So we do get signal that is biologically relevant. This is also translated into transcriptome signal. Here is a candidate gene that was identified. And yes, when we look in transcription data, we do get differential regulation of that gene. So great, we can find these things. When we are now considering moving from QTLs to something that's meaningful, we need elite-based donors. Yeah, no breeder is going to put a landrace into their population improvement program. With major genes and QTLs, forward breeding is an approach that's well established. And we can potentially use gene editing initially in validation of the predicted effects of alleles, and in the future in product development as that technology is more broadly accepted and deployed in our target countries. For polygenic traits, it's a little bit of a different category. And in maize, most things are polygenic. And we need different deployment strategies. And here we start to look at genomic selection that is informed by some of the outputs of environmental GWAS. So just here, don't look at the broad scale of data. What I want to show you here is essentially the outputs of breeding products using some of these technologies. In the white here, we see a series of experimental lines. These are new donors that we've been developing for drought. This material here in the yellow is the genetic check. This is essentially the elite recurrent parent that has been used. And these are two commercial lines that we use as checks. So here we're looking at treatments under drought and treatments under optimal conditions. Yeah, with the last thing we want to do is improve drought tolerance, but lower optimal yield. And as you see here, we do get a boost in all of these donors. So we are able to move polygenic variation forwards. It's not impossible. It's not simple, but it's doable. So here now we start to use environmental prediction. So we've all heard of genomic selection. Here we use genomic prediction. We're able to predict the accession and environment, the environment and accession came from with very high accuracy. And we're starting now to use this kind of paradigm to predict the potential of a landrace. What is the genetic potential of a landrace within a particular environment? These predictions, this is a nice slide that came out from Jeff Rossabara recently. We're able to use this information now to predict a genotype of an environment that may not have been sampled. So this is work looking at the landrace collection from Mexico and trying to say here, in this particular environment, what would the ideal genotype be? And then go back to the existing data and validate that. And this is proving pretty useful at the moment. So how do we use this knowledge in terms of pre-breeding? We use the environmental GWAS to identify novel variation, novel adaptive variation of breeding relevance. Sometimes we find large effect QTLs, they're very useful to move forwards. We're able, through comparison with the existing material in terms of the elite germplasm, to identify those alleles which are novel to the elite germplasm. The last thing we want to do in a breeding purpose is repackage something that's already been captured by breeders effectively. The prediction we use to get a rank order of those land races which may have higher value under a particular environmental stress. And we use this information together to identify those accessions which have the highest novel value for breeding. And these form the breeding starts for the development of new elite-based donors. And with that, at 45 seconds over time, thank you very much for your interest, and I hand it back to Marc.
I think it's very clear where we will go in the discussion. So now we go to the other talks first. And Rob Martienssen, here in Cold Spring Harbor, and also professor at the Howard Hughes Medical Institute. Please, Rob, tell us about epigenetics, where you are.
All right. Well, it's great to have this opportunity to tell you about our work on sustainability. I'm actually going to give the first of two talks on oil palm. Oil palm, of course, is one of the most challenging crops in the world to grow in a sustainable fashion. This is because much of it is grown in extremely sensitive environmental regions, as illustrated by this photograph, aerial photograph of Borneo, showing some of the oldest rainforest on Earth on the left, and an oil palm plantation on the right. Oil palm actually is one of the most important sources of edible oil in the world, vegetable oil. And in fact, human use and probably cultivation dates back at least 5,000 years to early civilizations in West Africa. And there are some other very interesting historical attributes associated with it on this slide. And recently, it's also been a major use for biofuels because of its oil composition. And all these things have increased demand for palm oil in the last few years. And my colleague from Malaysia, Ravi, will tell you a lot more about the economics and the sustainability in the next talk. I'm going to focus on what we can do with genetics and epigenetics to try to improve not only yield, but also the reproducibility of yield, with the intent of reducing land use, making land use far more efficient in these very sensitive environments. And we're going to start just by looking at some very classical genetic data. There are a whole variety of oil palms throughout Africa and E. guineensis, and early breeders discovered several traits that they recognized were actually controlled by single genes, although they were originally thought of as subspecies. Fruit color, for example, which I think Ravi is going to mention a bit more, is very important for ripening. The most important single gene trait is the thickness of the shell. This is oil palm is related to coconut. Think of that shell as being very important for determining oil yield. And finally, a detrimental trait, mantled fruit, which I'll tell you a lot more about, which turns out to be epigenetic. The shell gene, as identified by breeders in the 1920s, exhibits single gene heterosis in a very powerful way. So thick-shelled oil palm, known as dura, is homozygous for the wild type allele. It has a very thick shell. Thin or shell-less oil palms, known as passifera, are usually female sterile, but they don't have a shell at all, and this makes them very, very non-productive in all sorts of ways. But the hybrid, the single gene hybrid, tenera, has a much higher yield than the dura wild type form. And so breeders for the last 100 years, more or less, have been trying to make hybrids of tenera oil palm to get the highest yields. Okay, so tenera hybrids then have much higher yield and are the target of all these plantations of planting materials. The mantled fruit trait is a much more detrimental trait. This occurs naturally in some populations, but also in clonal palms, and I'll explain that in a moment. And mantled fruit has a very detrimental effect on oil yield. Shown on the left is a normal fruit bunch from oil palm, and on the right is a mantled bunch, which typically has no seed and is extremely low yielding. These turned out to be a big problem in clonal plantations, as I'll explain in a moment. More than 10 years ago now, the oil palm genome was sequenced in a collaboration between the Malaysian Oil Palm Board, as represented by Ravi, who led the project and who's going to give the next talk. Also Orion, a genomics company that I'm associated with, and our colleagues at the American Museum of Natural History. The new oil palm genome consists of 16 chromosomes and has a lot of features that are typical of plant genomes, and was released to the public 4 years ahead of publication in 2009. We could then set about using this genome to identify some of these key traits. And of course, the first to be targeted was shell itself, the shell gene. To do this, we took advantage of the wonderful pedigrees that breeders had developed. As you can see, oil palm has a pretty long breeding cycle, and these pedigrees started in the 1950s. And what we were able to do is identify homozygous passifera palms from these pedigrees. And we were able to use then a technique known as homozygosity mapping, which was actually first proposed by Lander and Botstein for human genetics back in the 1980s. We did this using sequencing. And shown here is just an interval near the shell gene. And you can see there's an awful lot of variation of SNPs between different homozygotes in these breeding pedigrees, except for the region that turned out to be the shell gene. And using additional recombination data from individual F2 populations that had been developed by breeders, we were able to identify the shell gene itself. It turns out to encode a MADS-box transcription factor related to the Arabidopsis seed stick, a D-class MADS-box gene. And we identified at the time just two alleles, one that had been used extensively in breeding for the last 50 or more years, the Avros allele, and additional alleles from African germplasm collected by the MPOB, the Malaysian Palm Oil Board. I just want to highlight this remarkable collection, especially after Sarah's wonderful talk. I think we have, an extraordinary resource here in Malaysia. The Malaysian Palm Oil Board has collected, through the efforts of Dr. Rajanaidu Nukia, over many, many decades, an extraordinary collection of germplasm from Africa. And interestingly, by mapping shell alleles to this collection, we're able to trace the origin of cultivated diversity to Ghana, to West Africa, which again is very, very consistent with what we know about the history of oil palm. So multiple alleles of shell were identified in this collection. And when they were sequenced, it's very interesting, they all fell in the same very small part of the MADS-box transcription factor, right on this alpha helix, which it's actually the DNA binding and heterodimerization domain of the shell gene. And this suggested, and I'm not going to go into all the data, but it suggested that heterodimerization with other MADS-box gene factors, which is well known in this family, might be important for yield. This actually fit very nicely with a theory that Jim Birchler had come up with years ago about single gene heterosis, in which the dosage of multimeric subunits of multimeric complexes, because of random association, had to be just right. And if you had too much of one or too much of the other, you would reduce the number of functional complexes. So actually, this fits Jim's theory extraordinarily well, and it's sort of fun to think about. So the shell gene was an extremely important trait. But breeders realized in the 1970s, and actually through the efforts of Unilever when they were involved in oil palm cultivation, that one way to enhance yields dramatically and very rapidly was to clone elite hybrids. So rather than having to cross them every time, which of course is extremely labor intensive and expensive, they realized that you could take, think of the heart of palm from an elite adult oil palm, which you already knew had fantastic yield, and then put it into tissue culture, regenerate plantlets, and get tens of thousands of genetically identical clones, which in principle should be able to give you much higher yield. However, very early on, it was realized that at some variable frequency, many of these cloned palms had the mantle trait. And this mantle trait, as I said, was extremely detrimental. It was frequently parthenocarpic and reduced yields, and actually really had a negative effect on the cloning industry, if you like. So that at the moment, only about 1% of oil palm, planted oil palm planting materials are clones. This trait was quickly recognized as being epigenetic because of the fact that it reverts, and that in crosses, it behaves actually, for those of you who know about it, it behaves very much like a paramutant allele. Early on, we realized, it was realized that the phenotype resembles homeotic mutants first identified in Antirrhinum and in Arabidopsis. And this particular homeotic mutant was called deficiens, in which the stamens, the male organs, were transformed into female organs, carpels. And this strong phenotypic resemblance was a clue to what might be going on in this epigenetic trait. But no mutation could ever be found in this gene, and so it was a mystery for a long time. So we employed an epigenome-wide association study, if you like. We've just had a great introduction to the genetic version. Using DNA microarrays because they could be used at very high throughput and low cost. So these are restriction enzyme-based DNA microarrays for DNA methylation. The wonderful, again, a collection from MPOB of hybrid materials which had been cloned and then are grouped into mother palms called ortets, and then either normal clones or mantle clones. So all three in this trio design should be genetically identical. And then we looked at methylation across the entire genome and asked for association of loss of methylation or gain with the mantled phenotype specifically. And this is what that EWAS plot looked like. It was pretty amazing. That's the entire genome. And there was just really one region that really stood out as having a strong association with the mantle trait. And this region, when we narrowed down, turned out to encode a transposon. Transposons, of course, were discovered here at Cold Spring Harbor by Barbara McClintock, but she also importantly realized, she called them controlling elements, because she realized they could control gene expression. And here was a great example of a transposon doing exactly that. The transposon was a line retrotransposon that was inserted into the intron of the deficiens gene. So this was a very, very strong candidate for the epigenetic trait. You might be wondering where this name Karma comes from. It was actually first identified in rice, and I'll show you that in a moment. But just a little bit more detail. bisulfite sequencing of the mantled, normal, and mother palms were revealed that there was in fact a differentially methylated region, or DMR, right on top of that transposon. And interestingly, it was only differentially methylated in one or two of the cytosine sequence contexts. So DNA methylation can occur either as CG, CHG, where H is anything but G, and CHH. And in CG methylation was unaffected in mantled palms, but CHG and CHH were dramatically lost from the CARMA retrotransposon. As I mentioned, CARMA had actually been identified first in rice, and it had actually been identified as one of only two retrotransposons that are strongly activated in rice tissue culture, so that made a great deal of sense. And it was actually called CARMA because it only transposed in the generation after tissue culture, in other words, in those regenerated plants. So it actually had CARMA, and so, which allowed us to, of course, distinguish the two alleles, the two epialleles, as either good CARMA or bad CARMA. And bad CARMA is associated with hypermethylation of the CARMA element. And I'm not going to go into any detail, but we actually published a paper more than 10 years ago now in Maize showing that DNA methylation can affect splicing. And in fact, there was a lovely paper in Nature just a couple of years ago from my colleague here at Cold Spring Harbor, Adrian Craner, who showed that histone methylation affects splicing as well. And I won't go into the details, but this graph just illustrates that in bad CARMA, aberrant splicing into the transposon truncates the deficiency gene and causes the phenotype. It's also associated with the loss of 24 nucleotide small RNAs, which guide this type of DNA methylation, and they're found in normal fruits, but not in mantled fruits. And also, they are sharply reduced in tissue culture itself. And again, I'm not going to go into the details now, but we think tissue culture loses this type of methylation. And we actually had a paper recently in Current Biology showing that it loses this type of methylation and loses small RNAs at the same time. And we think that this is what's responsible for the phenotype. Okay, so this led to my favorite Nature cover, Weed Out Bad CARMA, which illustrates the idea. And Ravi is going to tell you a great deal more about this in a minute. We think now that with a simple PCR test, we can predict the CARMA epigenotype, as well as the shell genotype and many other genotypes, before plantlets, shown here on the bottom left, are put into the nursery and into the plantation. And this allows culling of all of those low yield genotypes and removal of a lot of wasted land and an increase in land use efficiency, which is the goal of this work. I need to end just by showing the team. So this is Ravi, who's about to give the next talk. The Malaysian Palm Board led the genome project. This is the Orion company on the right, but we had a lot of help from many other genome institutes and sequencing companies in this whole endeavor, and they're all listed here. So thanks very much. I'll introduce Ravi. Ravi Sambanthamurthi led the oil palm genome project when she was the head of advanced biotechnology in the Malaysian Palm Oil Board in Kuala Lumpur. She now sits on many different advisory boards throughout the Malaysian oil palm industry and has been responsible for some of the main advances in biotechnology and oil palm over the years. And she's been my collaborator for a long time, and she's going to tell us about the impact of technology, biotechnology on oil palm sustainability.
Thank you very much. Thank you very much, Rob, for that very nice introduction. And not just the introduction, I've had the good karma of you giving a fantastic backdrop for my presentation. Now, I'll be describing the efforts in balancing environmental health and economic growth through improved productivity of the oil palm. The oil palm sector, as we know, is at crossroads. Large-scale expansion of oil palm cultivation has resulted in deforestation and loss of biodiversity. And this is especially so because, as mentioned by Rob earlier, the oil palm grows in highly ecologically sensitive regions of the globe. So, and the oil palm industry has also been blamed for displacing critically endangered species. However, we must also know that the biodiversity hotspots are also geographically coincident with poverty hotspots. And the oil palm industry has been a major vehicle in the socio-economic development of rural areas in this region. Oil palm industry has therefore transformed the livelihoods of millions of the poor, and recently it was reported that it has lifted about 2.6 million rural Indonesians from poverty this century. Although the oil palm is often associated with big plantations, it is very much a smallholder crop as well, with smallholders accounting for about 30 to 40% of global oil palm cultivation. The crop has therefore contributed a lot to social security. As mentioned as Sarah earlier, the global population is increasing, and it is predicted to reach about 9.7 billion by 2050. And this increase in population will be paralleled by increase in resources, including food and energy, and of course, vegetable oils. Now, the oil palm is arguably the best source of vegetable oils to meet the growing demand. This is because it is the most efficient oil-bearing crop. Planted on just 7% of the total land area occupied by all oil crops, it accounts for 36.5% of global vegetable oils. All other crops would require substantially more land to meet the demand, increasing demand for oil. However, it has to be acknowledged that the oil palm has to be grown more sustainably. And there is a need to decouple economic development and environmental degradation. And our strategy for this was via further improvement in land use efficiency by increasing yield and through value addition of circular economy mediated by investment in technology and international collaboration. We've already seen this. Rob has already introduced this. So as Rob explained, the shell gene is a very important gene. And the tenera, which is a hybrid of the dura, which has got a thick shell and therefore much less mesocarp. For your information, palm oil comes from the mesocarp. The oil palm has two types of oil, kernel oil, but what we know as palm oil comes from the mesocarp. So dura, because it has got a thick shell, has got much less mesocarp and therefore produces much less oil. And I will not go through the details because Rob has already given this, but importantly, the tenera produces 30% more yield than dura and is the commercial planting material in all of Southeast Asia. However, there is substantial contamination with dura, which therefore produces 30% less oil, and with Piscifera, which is female sterile and therefore does not produce bunches and therefore does not produce oil. And this results in a lot of loss in yield. Another important trait is fruit color, and the two main varieties of fruit color in the oil palm are the nigrescens and the virescens. The nigrescens is deep purple to black at the apex, and there is very little change in color as the fruit ripens. And therefore, the harvester who's at the bottom of the tree can't quite always tell when the bunch is ripe. This is quite different from the virescens, which is green when unripe and undergoes a rapid, profound change in color to orange red. And as you can see here, it's much easier to know when the fruit bunch is ripe. And this minimizes yield losses from harvesting underripe or overripe bunches. And therefore, this has an impact on yield since ripeness is a major determinant of oil yield. Besides the shell and fruit color, of course, tissue culture, as Rob has mentioned as well, that was one of our main targets for increasing the oil yield by basically mass propagation of elite material, right? But as karma would have it, the emergence of tissue culture abnormality put the brakes on large-scale cultivation of clonal material. And these are the different types of abnormality. And as described by Rob, the most common is actually the mantled. Now, we would not know whether it's mantled or normal until about 6 years when the clonal palms start fruiting. And therefore, this is high risk. And currently, only 1% of all palms planted are of clonal material, and emphasizing the important need for a screening test. Now, MPOB initiated the All Palm Genome Project for increasing productivity and, of course, looking for genes of economic interest. A very important resource, as Rob has also mentioned, for this expedition or for this activity was the germplasm collection. MPOB has the world's largest germplasm collection of oil palm. It carried out more than 40 missions over 4 decades and assembled more than 100,000 accessions of the oil palm. And this was an extremely important resource for our oil palm genome project. And we forged strategic alliances with different centers of excellence, including Cold Spring Harbor and Orion Genomics. And here, Rob has introduced this slide because he played a major role in this effort. And very importantly, having sequenced the oil palm genome, we could very quickly discover the genes we were looking for. That is for shell. It's already explained by Rob that it was a homologue of seed stick. And the fruit color, virescence, and this gene was actually an R2-R3 MPOB transcription factor, which was involved in anthocyanin biosynthesis. So when you see the nigrescence fruit, which is dark purple or black, it's because it's got anthocyanins. But the virescence fruits lack the anthocyanins, and therefore you can see the chlorophyll changing then to carotene accumulation as it ripens. And so it's very, very important because, as I said, fruit ripening is an important determinant of oil yield. And I will not go too much into clonal abnormality because Rob has done such a fantastic job. But importantly, we discovered the mantled gene, which was deficient, and of course, the karma epi mutation. Now, we published these, of course, but more importantly, we translated these discoveries and they were commercialized as the world's first high-throughput genome-based diagnostic systems. And the technology was licensed to a Malaysian company, Orion Bioscience, and the technology was trademarked Sure Sawit. Sawit means oil palm in Malay. Okay? So there were 3 series of Sure Sawit technologies that were developed with Orion Bioscience. The Sure Sawit shell for testing for shell, Sure Sawit ver for virescence, that is for fruit color, and Sure Sawit karma to test for the risk of clonal abnormality. The test can be done as early as in the nursery stage by just taking a leaf bunch, or even earlier at the seed stage. And after analysis, one just has to plant the desired palms and leave out the undesired. I cannot overemphasize the importance of testing before planting. The oil palm is a perennial crop with an economic lifespan of 25 years, which means that what you put on the ground today has a bearing on sustainability for the next quarter century. Okay? Now, this technology also spawned a whole new biotech industry in Malaysia and offering employment for local scientists. This is Orion Bioscience, the central testing facility in Kuala Lumpur, Malaysia. We also have DNA extraction labs in Sabah in the island of Borneo, as well as in Indonesia. Now, the Sure Savit was selected by the BBC as one of the 8 most disruptive technologies in the world and was featured in BBC's Digital Disruptor series. Here you have Dr. Rajan Naidu, who led most of the expeditions to collect the wild oil palm. Using the Sure Savit shell, MPOB tested more than 1 million seedlings, representing about 93.5% of the oil palm planted area in Malaysia. We were surprised to find that 12.8% of the palms were not the Tenera type that we expected, but they were undesirable low yielders. And we determined that 1/3 of the sites would see a 12% increase in land use efficiency with DNA testing. On average, the entire planted area in Malaysia would see a 6.9% gain in yield and land use efficiency. And this translates to about 450,000 hectares. And this is Malaysia. If you assume that Indonesia and other producer countries have the same level of contamination, you would think like in Indonesia, you could be saving more than 1 million hectares of land. This is a recent study of the oil palm plantations in Indonesia, and it was reported that there was a 38% gap in yield and land use efficiency overall in Indonesia. And these pie charts, the red section shows the gaps in the land use efficiency. This slide superimposes the Malaysian scenario with DNA testing. And you can see that land use efficiency gains by DNA testing. So basically, DNA testing is a very powerful tool to increase yield and land use efficiency and sustainability. Our next strategy for increasing productivity was value addition through a circular economy by harnessing byproducts of oil palm processing. Using the processing of palm oil, after the oil is extracted, the non-oil components enter the aqueous stream and are discarded in the waste stream as palm oil mill effluent, or POM for short. Now, Malaysia alone produces about 58 million tons of POM. And this can be a huge environmental bioburden if not treated properly. While there are very stringent regulations in Malaysia for disposal of POM, it obviously adds to the cost. But it occurred to us that in our zealousness to squeeze out as much oil as possible from the oil palm, we had lost sight of the fact that the oil palm is in fact a fruit, a very colorful fruit. And colorful fruits like berries are high in polyphenols, which have numerous health benefits. It thus occurred to us that these polyphenols, which must be present in oil palm, are going down the drain into the effluent pond because they are water soluble and enter the aqueous stream. We tested the POM, and indeed we found it had significant amount of polyphenols and other phytochemicals like water-soluble vitamins. So this presented a great opportunity to turn around a problem to add value downstream. We designed a very simple solvent-free process to extract the polyphenols from the aqueous stream before it is discharged as POM. And we also built a pilot plant to produce enough of this material for us to carry out pre-commercial and preclinical and clinical trials, as well as pre-commercial trials. Together with our collaborators across the globe, we confirmed multiple biological activities of oil palm phenolics. So it is pretty obvious that this innovation has got economic, health, social, and environmental impacts. We commercialized the technology and licensed it to a company known as Phenolis, which is registered in Malaysia, the US, and Mexico. And I'm very happy to say that the world's first oil palm phenolic production plant was built in Mexico and started operation in January of 2019. I had the opportunity of going there, fortunately, just before COVID struck. Okay. These are some of the products that have been produced. The first batch of commercial products, trademarked palm fruit bioactive complex, was rolled out as a bulk ingredient in the US in June 2019. And a wellness company known as Amari Global launched the first product formulated with oil palm phenolics the same year. And oil palm phenolics emerged as one of the finalists in the Nutra Ingredients of USA Awards 2019 as an ingredient for healthy aging. I have many people to acknowledge for our success in our work. Very big thank you to the current Director General of MPOB, Dr. Ahmad Parviz, who has been so supportive of our work. Dr. Arif Manaf, the current Director of the Advanced Biotechnology and Breeding Center, who's also a team member from the genome team. And these projects have spanned several years because there was a lot of, obviously, a lot of thought process went into it. And it's many years, so we've got several ex-DGs to thank as well. Now, our success is mainly attributed to teamwork across the globe by amazing teams. I have to use this same acknowledgement slide that Rob used. And I would just like to, I cannot mention everybody, but this is Rajinder, who is the head of the genomics unit, Dr. Melina Ong, the head of the breeding and tissue culture unit, Dr. Leslie Lau, the head of the bioinformatics unit, and a whole wonderful team. And here you've got Nathan Lakey, who leads the Orion Genomics team, and they played such an important role in our success with regard to the Genome Project. And of course, you've got Rob Martienssen, who injected so much knowledge and wisdom that contributed to the success of the Genome Project. And for the phenolics team, I'd like to acknowledge Dr. Said Fairuz, who carried out clinical trials, my ex-PhD student, Dr. Liao Sun Sen, who carried out a whole range of gene expression studies to show the multiple bioactivities of oil palm phenolics. I would especially like to thank Dr. Tan Yu Ai and Dr. Kalyana Sundram, my friends, with whom I shared the idea about oil palm phenolics in the effluent, and together with MPOB, we built up a whole team with international collaborators, and we continue to collaborate till this day. With that, I thank everyone.
Beautiful, the progress that can be made if the molecular techniques come together, and that you can see that classical oil palm breeding is one thing, but what can be made now? But I'm afraid for the discussions, there will be some questions. Oh, I'm sorry. I thought I was unmuted by. Okay, thank you very, very much. I'm afraid you didn't hear me when I was talking on the progress that the molecular science coming together with the breeding and all the efforts that farmers there had done till now, that the progress gives a lot of hope. So for the discussions, I think the panelists can start, but of course, I take the privilege of being the first one asking, it's good to see how you can work now with the elite germplasm you have and how you can make better use of it. But would it not be also worthwhile that you would join the action that some molecular work would be done to create new varieties with all the elements that you have now found, that by saying transgenics and make it in a way that you keep a maximum the elite variety, add some traits, because 25 years is still a lot of work and a lot of time before you harvest it. And for environment, your work shows how urgently it is for the whole area of Malaysia and Indonesia and other parts of the world to have high yield, so that you really use less land, and that this land can go back to well, wild forest for biodiversity, all the problems that some scientists have identified now, and that we want environmental changes for it. So doing that, I think it's also urgent that the research on adding or altering sequences also can go on. Who wants to take that up?
I could say a couple of things right away. I mean, you are, of course, exactly right. I mean, we've identified, just by looking at natural variation, very, very similar to Sarah's work with maize, lots of opportunities for genome editing that would allow us to immediately introduce beneficial traits into elite germplasm. But at the moment, genome editing is still not, it's still classified as GMO. And as long as that's true, export crops such as palm oil, or export products such as palm oil, are going to have a hard time being able to use this sort of technology. I think it's really important to keep lobbying, especially the Europeans, to allow or change the regulations on genome editing. But as you could see from our talks, I mean, there are so many opportunities, and it will greatly accelerate most of the breeding schemes we have. So I think you raised a really, really important point.
Yes, but I'm afraid that lobbying is not enough, because that's 30 years of lobbying that was done by just argumenting with scientific arguments. And the answer we receive show us that the product is there, and then we will judge, like we do for all the products that material sciences has made through really all the possible scientific approaches that you can do there. So they say, show us, and to show us, we should at least obtain, and I think in Malaysia and Indonesia, it would be perfect that if authorities say, at least we go for this work to make the prototype, okay, it can take 10 years, but at least it has been done. It's now, there are so many new things that you will identify through the molecular techniques. You have now 2 traits that you can follow, but we know the way epigenetics is going, that we will understand better the gene expression in plants. And at that moment, if we do that, we will do a lot of alterations that will make better stocks for starting, better germplasm. And a better germplasm you can make in greenhouses for Arabidopsis, but for oil palm tree, you have to see the real facts. And that should be possible to communicate. So I think it's an enormous task for the younger generations to say, we have to explain what is the science. And if you claim that there is any danger, show us, explain why you see that there would be danger.
If I could just add to what Rob said, and I agree. Well, although commercial palm oil is GM-free, but we have always believed that the science must go on. And MPOB actually has got work. It started many years ago on actually producing transgenic oil palm. But in this case, the target was more for tailoring oil composition. So we have that project, and we have putative and transgenic oil palm in this greenhouse, but not for commercial purposes. And there is also work now on genome editing, also for oil composition. and other traits like resistance to Ganoderma. I think there was a paper by the Indonesians on this. We're not sure if they've demonstrated resistance, but there is work on genome editing for Ganoderma resistance. Ganoderma is the most serious pathogen of the oil palm. So work is going on. Because as you say, the oil palm is 25 years and we really have to start now. So the science is going on, and perhaps by the time the EU and the rest of the world are accepting genome editing, I think MPOB would hopefully have the science all worked out. Yeah. Mm-hmm.
Yeah, but it's indeed, if you just do the science, you already can convince a limited number of the populations, and there you will have persons that find the right words to explain it to society. But it has gone. Well, I don't want to mobilize the talks, please, among the panelists. What ideas come up for you, among yourself, that your science is bringing, and that should come up, and that the society should know that any, or should I go on doing questioning?
I can see we do have some questions.
Yeah, I can actually get started with a few questions. I have a few of my own and a few came in from the audience, but let's start with one from audience member Doreen Ware. This is directed to Sarah. She says, beautiful work and talk. Based on your initial screen of the environmental factors using the GEPV, how many breeding cycles are needed to get germplasm that breeders are willing to use in their programs?
Okay, very, very good question, Doreen. So the current number of generations that we've been able to really achieve a product that is semi-elite, that's uniform, that breeders are willing to use is as little as 4 generations. So that, as we can in the field here push easily to three cycles a year is pretty fast. The challenge has been, up until very recently, we haven't been using and leveraging a lot of the genomics in that. We have been doing a lot of field phenotyping, which we can only do once a year. So that limits the time, because we have a certain periodicity that we've got to adhere to. So the products that have come out now have been developed across about, let's have a think, 7 years of research efforts. Now, as we start to move these things more into a parallel structure, and rather than the classic, we will develop a population to really understand what the biology is and to start to dissect that more cleanly at the same time, or earlier than developing populations that breeders will actually use, we're combining these things together. So we're discovering as we are breeding. Nurseries themselves are cheap and easy to make. Yeah, making crosses is not difficult. Making the progeny is not expensive. The challenge is in the phenotyping of that. And the cost benefit really is how quickly can we get a product to breeders. So we're starting to run these things in parallel, phenotyping and validating as we move forwards, using the understanding that we have to select the higher value starting materials and the higher value alleles to focus on as part of that process.
For the corn and wheat problems, there is so much work going on all over the world. I think it also should be very important that you can come up and really explain how the environmental advantages are for using these varieties, and for particularly for the areas like Africa, where they cannot at the moment, through the history of the country, develop the science themselves, but all scientists are in their solidarity could build up together efforts. Like in the beginning of the CGIR, ideas were there, but now the tools have progressed so nicely that you can do demonstration plots and in well contained conditions that people should be really convinced that the science contributes to the environment, and that in the timing you do it, that it will be much better than making rules of we have to do mitigation for the CO2, and we have to wait till the whole industry reconstructs. We can say that I'm convinced we have with this and this time scheme we can get positive results with the science we are doing. Can you convince at the CG system that this attitude should be taken?
I mean, the CG system is science driven. Yeah, we use science as a guide.
I'm convinced.
But we have to, within that context, respect national laws, and we do partner with varying countries in transgenic and gene editing products. We've got a number on the way for both maize and wheat, at least from the CIMMYT perspective. But obviously we do that only where and when a country has requested use and development of that particular approach. That said, across the CG now, we have a number of collaborations with groups like IGI at Berkeley, where we're exploring the use of technologies like gene editing for a variety of approaches. And one of which I mentioned is the validation. We can identify what we think may be a high value trait in a wild relative, in a landrace. That takes a lot of effort to get into elite germplasm. Now through substitution and other technologies, we can quickly validate the effect of that in an elite background, see if it's really worthwhile. Now that product can, if there is the appropriate legislative framework, be deployed in partner countries. Where there isn't, at least it gives us information that this particular allele or set of alleles are high value, and we need to start developing that pre-breeding process. So through the use of this technology for validation, we can increase the familiarity of different stakeholders of the technology and let them make their own science-based decisions in terms of what they want to adopt nationally.
Yeah, I'm convinced absolutely that you can do, and I consider that as well, at the moment, in despair, the only thing that can be done. But you could think in the way, if there are unknowns, and for all society problems, there are so many unknowns where in the human sciences that we don't know what is better, this and that. And we also have to make decisions. So explain that the product is there and really taken a leading position in demonstrating that you can make the product and challenging them with the arguments that they are considering as most valuable. You cannot explain you want more yield because that is an economic political system. But you could explain for environment, if you want to have these goals, if you could make this, then it will take you only so many years or such time to have that in a large scale. I think that is waiting which organization will take it up and explain it with all respect for why people are afraid and don't understand this type of arguments. That is sure that we have to do that. That's the way our society works. But if you don't go on explaining it and say that how urgent it is, and that you see what is the problems with overpopulation, what is the problems of poverty, what will your society if you don't change that see with environment that goes on, and use the arguments that could maybe convince the major stakeholders in society.
All right,
Thank you, Marc and Sarah. The next question, this is for Ravi and Rob. So first of all, very interesting talks on the impact of genomics on sustainability. Now, you probably agree that for this approach to succeed, having a complete and well-annotated reference genome is critical. Now, this is possible with technical advances in sequencing techniques, but this could still be a biological problems for complex genomes like, polyploidy or duplications and such. So could you please speak to this challenge and possible ways to overcome this?
Sure. I mean, if you don't mind, Ravi.
Please.
Absolutely. I mean, the oil palm genome was actually done with a combination of next generation sequencing and much more old-fashioned sequencing as well. And these days, for example, polyploid genomes are completely accessible. We've recently in my lab, we've just recently sequenced some polyploid aquatic plants, and you can really phase genomes incredibly well now for very low cost. And I think this really opens up opportunities for agricultural biotechnology in ways that weren't around before. There are also new ways of epigenomic analysis, which also allow very rapid. Again, we use some pretty old technology with the Karma discovery, but the possibility to do that now is much greater. I noticed there was a question from Sebastian Marc as well on how much other plants, other crops could benefit from this sort of approach, and I think it's going to be huge. There's a lot, for example, there's a lot of plants that are micropropagated by tissue culture. The biggest, interestingly, is actually the orchid, which is literally tens of millions of clones of orchids are propagated every year, and this sort of technology would be very useful for orchids. Orchids have huge genomes which were completely inaccessible until very recently, and now they really are. So yes, I think this holds a lot of promise.
So Rob and Ravi, if I can just follow up. Murali Murugavel asks, what is the water use efficiency of oil palms versus other plant oil sources such as soy? And then a second part to Murali's question is, can oil palm plantations actually grow in land-rich locations like Canada or Australia?
First question is for the water use efficiency. Well, we haven't really done a comparison between soybean and oil palm, but the fact that the oil palm is a perennial crop and it's got a very deep root system. So I believe that the water use efficiency is actually is better than soybean. It's a perennial crop with a good root system, right? As for the question on whether it can be grown in Canada and other land-rich areas, no, as I explained, it's because the oil palm grows in, I think, best 10 degrees north and south of the equator in the highly ecologically sensitive areas of the world, unless you come up with something that's that can grow at different temperatures, very unlikely that you can grow it in Canada simply because of the temperature, or Australia. I think there is some oil palm in Australia, though, in the warmer areas. But generally, the question is no, I don't think so. Rob?
Yeah, I mean, we have occasionally thought about not just oil palm, but things like sugarcane, changing. We can certainly do a lot with flowering time. We understand that extremely well now, and photoperiod dependent and temperature dependent flowering. But having said that, that's not the only thing. And oil palm especially seems to be very sensitive to a lot of climatic variables that are not fully understood.
Yeah.
So it will be a challenge to engineer some of these crops to grow in other places. It's amazing how maize has been able to
oil palm solidifies at low temperature.
Sorry,
There was,
did I hear? Couldn't quite hear.
I didn't hear that.
Plus also another thing is that if you plant it in the cold, because the yield will be low, but we also find that actually the oil palm has got a cold activated lipase. So if you're going to grow it in cold climate, there's going to be a very high free fatty acid content, and that's what produce low quality oil, even if you can produce. So there are many, many things that you have to sort out if you're going to try and grow it in a place like Canada. No, even if it can grow at low temperature, there would be an effect on lipase and things like that. So I think the answer is no. Mm-hmm.
Thank you, Ravi and Rob. Sarah, here's one for you. You talked about a nice approach, a data-driven approach, combining native genetic variations with principles of modern informatics to identify elite breeding lines. We heard about this approach with respect to maize. It'll be interesting to see how this approach has been translated to other crops. And if so, are there any challenges?
Good question. So obviously the first challenge in this approach is having collections that have good georeference data. We have recently looked at the wheat collection of the CG, which is well over 100,000 accessions. Only about 6,000 of those at the moment have good collection site information. A lot of this information got lost as it's been translated from the paper records into the digital records. So we're going back and pulling out this valuable information now. So that's the first thing that you need to have. The second thing among those collections that have georeference data is sufficient representation of environmental climes that you're interested in. If most things have come from environments that are particularly uniform, at least across that collection, you're not going to get particularly good return of signal. And then the question is, what is the cline in terms of FSTs across these materials? Maize is particularly good. We get very gradual shifts in allele frequency, so we're able to pick out a little bit more nuance in the analyses. That said, we've been looking at proof of concept of this in other species outside of CIMMYT. We've got good proof of concept in sorghum now, in cowpea, in bean, and we've got a number of projects just getting off the ground now, funded by a number of international donors. So we're expanding the range of crops that we're applying this to about 9 different species now that are some of the core fundamental species for at least human nutrition in the global south.
Thank you.
All right, I have one last question before we conclude this portion of the program and move on to our Meet the Panelists session. Excuse me, and that is, can you guys elaborate a little bit on some of the major challenges in translating genomics research, say in oil palm and maize, to commercial outcomes? I'm sure there are many, and we can have a whole discussion on this, but if you could just tell us a little bit about some of these challenges.
Sarah? Maybe I'll have a go then. As far as the oil palm is concerned, obviously DNA-based technology involves high startup cost. And I'll just talk based on the SureSAVIT series anyway. It involves high startup costs. So that's obviously a major challenge, but it wasn't the most important challenge as far as commercialization of the SureSAVIT series was. And I think one of the problems we have is that the oil palm is a perennial crop. And if you plant it, you don't see the effect till 6 to 8 years down the road. And so there's sometimes a reluctance to do things differently because you're not going to see your results till from 6 to 8 years down the road. So while many oil palm companies are very keen, but you also have people who feel that, well, we're not going to see it while we are the manager, like you say, the managers of these companies, their KPIs are based on what? Short-term performance. They're not going to wait for 8 years to see the difference. So I think there is some reluctance in some parties to implement this technology. So adoption can be a problem because of the perennial nature of the crop. You don't see the result straight away. It takes about 6 to 8 years before you see something. So that's one of the challenges we have as far as translating the genomics work to, let's say, to do a routine DNA testing. Yeah.
And if I take that further, I mean, we see some of the same challenges in the annual crops. I think one additional challenge we have, and in particular with maize, it being such a well-studied species, is there's a lot of the time a disconnect between what are the real needs of the breeding programs and what things are actively being researched on now. So lack of talking to breeders, talking to market specialists, and everybody has their favorite trait. And in some cases that trait is super interesting from a biological point of view, really understanding the mechanistic basis of gene regulation, but it's not something that the breeders need. Flowering time is a great example. We've got millions of studies on flowering time, which is super important. Every breeder I know working on maize says, I can measure that in the field really quickly. I don't need DNA markers for it. So it's trying to identify what are those key points that are really valuable to breeders and are really valuable biologically, and making sure we use those as proof of concept examples to drive technologies forward. Because there's a lot of things that sit on the shelf and get caught up in more hype than anything. So it's really identifying those high value priority cases and moving them forwards effectively.
Yeah, I can only add to that by saying that I think just from the purely technological point of view, agriculture and sustainable agriculture has benefited enormously from developments in human and biomedical research. So sequencing technologies, diagnostic technologies, all of that, and the very high throughput sciences we can bring to bear. So in a way, at least for agriculture, the real rate limiting step is in regulatory and market approval. And that's sort of, it's out of the hands of scientists in many respects. And I think Marc brought out some really, really important issues there that we need more communication between the biotech sector and these regulatory bodies, more understanding and in the general public as well, but also with breeders, with seed companies. The industry is often not, certainly for oil palm, it's often not vertically integrated in the sense, especially smallholder farmers, they're only responsible for growing the crop. They're not responsible for making the seed or even for extracting the oil. And so, they only get a piece of the value chain. And it's therefore difficult, even if they're going to end up being the ones who benefit the most, it's difficult to, persuade an entrenched, a system that works well to change their ways in order to benefit the people who matter most. But that is the challenge.
Great, thank you. So at this point, I think we will close out this portion of the program. Sarah, Rob, Ravi, and Marc, I want to thank you all so much for being with us today, for some really great talks on your work and discussion on what really is a very critical and globally relevant topic. I do want to remind all of the trainees to please stay on the call for another 15 minutes or so for a special Meet the Panelists session. And for anybody who can't stay on and won't join us for that session, please click the survey link in the chat box and give us your feedback on the series. A big thanks to the audience for joining. We look forward to seeing you again in 2022. Keep an eye out on our website at lifescienceacrosstheglobe.org for more details about the next set of topics that we'll address next year. Thanks, everyone.